My name is Harry Mu. I'm gonna interview Professor McElory. My partner and I read a paper about the reduction of the population of the winter plunders, which really interests us. But because there are many difficult terms in biology, so we don't totally understand it. So can I ask you some questions?
Of course.
First, I want to ask a very simple question. What involves you to study the toxicology, the aquatic toxicology?
Okay, well, since I was your age, a very long time ago, I've always been interested in how marine organisms interact with chemicals in their environment. And so I've, for many, many years, have studied different types of marine organisms, many that live in close association with the bottom. So many of the chemicals I work with are hydrophobic, which means they tend to accumulate in sediments. And so I specifically look at how animals that live on the bottom or even live in the bottom sometimes. I've worked with worms and clams that actually eat and live in the mud, as well as a number of fish that either bottom feeding fish or like the winter flounder, the fish in this study, they bury themselves in the mud. So they're in close association. All their surfaces are covered by mud, and they eat other little animals that live in the mud. So they are exposed to a lot of the chemicals that are found in the mud. And the other interesting thing about these winter flounder is that the female fish lay eggs, fish embryos, that stick to the bottom, so they're exposed to the mud. And the baby fish, the juvenile fish, live in the same area. They don't swim very much. And so they're exposed to chemicals in that area for their first 2 years of life. So unlike many fish that are called pelagic fish, they live up in the water column and they swim all over. With a benthic fish like these winter flounder, you can be reasonably confident that they have mostly seen chemicals in the area where their embryos were born. So this is the most important question. The reason for you to study the winter flounders, because they are they're living in the range, so they are influenced by the polluted water or something else. Yeah. Right. So with marine organisms, particularly marine fishes, one of the concerns is if they migrate a lot, you don't know where they picked up what may be affecting them. And with these flounder, you have a pretty good idea whatever is affecting them is due to where they live.
So it's easy to study.
Yeah. So I started working with winter flounder actually back when I used to teach in Boston. I worked with adult winter flounder there, in part because early studies done by other people had shown that in some places they seem to have a high incidence of cancer, neoplasia. And I was trying to see how they responded to a group of chemicals in the environment called petroleum hydrocarbons, some of which are known to be carcinogenic. So I did that work in Boston. And then when I moved down here to Stony Brook, I was involved in another project looking at chemicals coming out of sewage treatment plants. And we showed that there were fairly high concentrations of chemicals that have been shown to feminize fish. So they act like estrogen. They're estrogen mimics. And so being familiar with winter flounder, so we found this area near Kennedy Airport in Jamaica Bay that had high levels of these feminizing chemicals. And so I hypothesized that if these feminizing chemicals are being released in Jamaica Bay, and it also turns out they are particle reactive, which means they end up in the sediments, that sediment-dwelling fish, like the winter flounder, might be a good place to look to see if there were effects of these chemicals in the environment. And indeed, we showed that in fish from Jamaica Bay, that they had a very high incidence of female-to-male fish, many more females than males, and that these juvenile fish, which aren't yet, we don't really know if they're males or females yet when they're that young, that they were producing higher levels of a female-specific protein that normally only female fish, once they're adult, are producing. Yeah. Well, we didn't know for sure with the young fish whether they're male or female, but we did know, compared to a reference area out east on Long Island, in Shinnecock Bay, that these fish from the sewage-impacted harbor had higher levels of this female-specific protein called vitellogenin, which normally in adult female fish helps lay down the egg yolk for the eggs. That's what its role is. So this was work I did some time ago. And the study that was written up in this paper was work done more recently, where we tried to look at not only fish from Jamaica Bay, but fish all along the south shore of Long Island, 6 different sites, to see if there was any link between exposure to these types of chemicals and things that would be important to the fish population being healthy. So we collected fish, we looked at mortality rates. Young fish, it's a very tough being a young fish. High mortality everywhere, that's normal. But what we were asking is the mortality higher in the areas with the more contaminants? Do they grow faster or slower? Are they plumper? We looked at something called a condition index that measures, it's a rough measure of your length to how much you weigh. And if you're plumper, you have a higher condition index, which should indicate that maybe you're getting enough food to grow. So we looked at that. And then we looked at expression of a variety of genes that are associated either with contaminant exposure or immune response or energy status.
This turns out, I think it's very important in the paper, but because we don't really understand this words. So can you explain this more?
Okay, so what we did in this paper, we collected fish from all these areas. We measured these other measures of biological response that mostly were reported in other papers, so not in this paper. In this paper, what we reported primarily was data on gene expression. So what we did is with these fish, when we caught them, we immediately froze them very quickly between dry ice. Yeah, so two blocks of dry ice and a flat fish, instant freezing. Then we keep them frozen and we put them in the -80. And so they're basically completely in suspended animation there. And then when we get ready to analyze them, we very quickly, we very slowly warm them up, keeping them on a block of ice, which allows us just enough that we can cut into them, because when they're frozen hard, you can't cut into them. They're hard like ice. So we warm them up just enough that we can cut into them, and we take their livers out and start processing them to extract the RNA out of the liver. And by being very careful how we thaw them, we're basically able to get the liver out while it's still frozen. And that produces very high quality RNA. And so we extract out the RNA. It's a simple chemical extraction. And then we turn that into back into copied DNA. So we use the template bases in the RNA. We turn that into what's called complementary cDNA. And then using known sequences for different types of genes, we make we use something called polymerase chain reaction. Have you heard about PCR?
Oh, I haven't heard of it.
I'm sure you've heard of it. What it uses is this. I can explain this easily. It uses a set of bacteria that have very set thermal tolerances to amplify, make copies of DNA.
Oh.
And you use these bacteria, and you use what's called a primer. So it's a sequence of DNA that targets a specific part of the DNA related to a specific gene, like say maybe the gene for brown eyes, or the gene for vitellogenin, which is this feminine specific marker that we saw earlier in fish. And knowing the sequence of those genes, you develop these primers that target one end of a part of the gene and another part, and that is called a forward and reverse primer. And then through help of these thermosensitive bacteria and the different oligonucleotides, you can build and identify a sequence in your sample that has that portion of the gene. And it's called amplification, and you're usually building on something around 200 base pairs of sequence. And so knowing some sequence information about the genes, not necessarily even in your own fish, but maybe in a related species, and all this is available online. You design these primers, and that allows you to identify in your sample whether that sequence is present and how much of it is present. And the technique used to do that is called polymerase chain reaction, because it finds it once and then it keeps finding it again. And there is a modification of that sticks a fluorescent probe onto every time it makes one of these little, recognizes one of these sequences. So you can actually, as it keeps making copies of this, you can find in a machine that not only, it's called a thermal cycler, it goes up and down with these temperatures to make the copies, but it is also a fluorescence spectrophotometer at the same time. And so you can actually see the fluorescence in your sample building over time. And you do this in little 96 well plates. I'm sure you've seen those. They have like 200 microliters of fluid. So you can run simultaneously these reactions for like 80 some samples at the same time. And this fancy spectrophotometer every so many seconds measures the fluorescence in each one of those wells. And so over the course of about an hour run, you get increased fluorescence only in the wells where it's amplifying the portion of the gene that you're interested in looking at. So we did that on individual fish from all these different sites, and what we found is that a lot of variability, these fish are wild fish, so they're not clones of each other. They're not, and they're responding to everything that they see in their environment. We had a hypothesis that the fish from the more polluted sites, which are Long Island or sites further west, that we might see reduced condition. Maybe they're not as plump as the fish from the east. That we might see some evidence of some of these genes involved in immune response, the ability to fight off parasites and infections, that might be reduced. Or that it might be enhanced, actually, because if they're seeing more pathogens, maybe they would have, they would be more enhanced. And that we also hypothesize that we might see some of the genes we looked at were ones that respond to the presence of different types of contaminants in the environment. We looked at a gene that responds to estrogen-like contaminants, and another one that responds to more petroleum-like contaminants. And what we saw was not necessarily what we predicted. We didn't see clear east to west patterns, either going up or down in many of the genes. We saw site-specific differences, but they didn't always line up with things being worse in the west and better in the east. But what we did see, looking at the expression of all these...
we have 20 minutes. So
oh, okay.
So can
Yeah.
I ask? So
Yeah, we'll go back.
Questions
Uh-huh.
that we are really interested in. Because you have mentioned that the reduction of the population is mostly caused by the overfishing and the change of the environment and the reducing habitat condition, which are all, I think it's all caused by human. So, and also in my hometown, there are many fish in my hometown, but the water is polluted. So can I ask you, what do you think about the relationship between human and other lives? So,
well, certainly there are many environments, both on land and in freshwater and in saltwater, and unfortunately many in our home country, where pollution is causing problems. We live in an area here where there are a lot of people, but still a lot of wildlife and a lot of fish, but probably many less fish than 50 years ago, 100 years ago, for sure. If you read the old reports, fish were much more abundant. And part of that reduction in the number of fish is because we have caught so many of them for eating or for fish food. But we think part of that is due to the fact that we've polluted the habitat with chemicals, with nutrients, and this study was designed in part to try to identify which factors may be associated with population decline. We know from other studies following the numbers of these fish in this area that there's been a very precipitous decline in their population over the last 20 years. And a related study we did on these same fish showed that their effective population size, which means the number of parents that have given rise to these little fish that we measure, is extremely small. Less than 1,000 fish, we think, contributed to the fish we caught in this study, and that is extremely small. So we think these particular species, these winter flounder, in these coastal bays, is been diminished so much by early over-harvesting by fishermen, which isn't really going on anymore because there's so few of them left, and potentially degradation in their habitat, that it is really on the verge of maybe what's called local extinction, where we would not see any more of these fish in these inshore waters. The data we got in this study suggested that there is a strong relationship between, or at least a statistically significant relationship between exposure to these estrogenic chemicals, immune suppression, and at least one of the energy genes that links to their growth rate. So what we've proposed to do to follow up in this study is to try to do laboratory experiments to try to figure out which of those factors are more important. And we don't know yet because we haven't done those studies. But it's not like, I think, to answer your question, any one of those things alone may be responsible for the demise of these fish. We think it's actually a combination.
Oh, thank you. Thank you for your things.
Oh, you're welcome. I hope I didn't talk too much. As you can see, I'm very excited about this work, so I tend to talk too much.
Thank you.
All right.
Thank you.